Now that we've talked a little bit about how cells divide and that DNA is able to replicate, be transcribed, translated, and produce proteins, we can talk about how these processes give rise to what we call the field of genetics. This involves talking about four major topics. The first one is the basics of Mendel's law. This is probably what you already know about how genetics works. Then we'll talk about variants on Mendel's laws, things that don't fall into two nice categories of recessive or dominant. We'll talk a little bit about the chromosomal basis of inheritance, so we'll tie it back to mitosis and meiosis, and then we'll talk about the difference between sex chromosomes and sex-linked genes. So let's go ahead and start with Mendel's laws. Heredity is the transmission of traits from one generation to the next. Genetics is the scientific study of heredity. So we know that things are inherited from our parents, but through genetics we want to understand why and how. The first person that is largely attributed to being the first geneticist is Gregor Mendel. Mendel's experiments involved crossing pea plants and tracing what he called traits. Traits were things that you could see about the plant, what color it was, how tall it was, and then he would follow those traits from generation to generation. He hypothesized that there were alternate versions of traits, or genes, and he called these alleles. Then that they determined whether or not something was heritable and how. The basic setup of a genetic experiment is as follows. You start with two parents. In this case, we have a purple pea plant and a white pea plant. This is called the parental generation, or filial generation 0. You then cross those individuals together to create progeny. This is the first generation, or the filial generation, F1. In this case, in this version of the experiment, all of the plants in the F1 look like the purple parent. Then you take it one step further. You take two individuals from the first generation and you cross them together. This creates the second generation, or the F2. In this case, we see that 3 of the plants are purple and white has returned. The reason that pea plants were such a good choice is that they are complete flowers. This means that they contain both a female part of the plant, the carpel, and the male part of the plant, the stamen. If you take the stamens and remove them, now the plant can't fertilize itself, and you have control over who does the fertilizing. In this case, we have a female purple plant, and we're using sperm from the white plant to create the cross. We then get that carpel to mature into peas. And the peas are very easy to plant and observe the offspring. From his research, Mendel came up with 4 hypotheses. We'll describe them below. Your book lists them as very long, but we're going to also give you a shorter version of what they mean. The first one is there are alternate versions of genes. We call these alternate versions alleles, and that they account for variation in inherited characteristics. So for example, you might have a gene that specifies the color of your eyes. Some people have alleles that suggest that their eyes should be blue, and some have alleles that tell their eyes to be brown. The second hypothesis is that for each characteristic, an organism inherits 2 alleles, one from each parent. This makes sense given what we know about meiosis and fertilization. Eggs and sperm are haploid and come together to make a diploid organism. You get 1 copy of all of your chromosomes from your mother and 1 copy of all of your chromosomes from your father. This also means that alleles have to have a relationship with one another. For example, an organism that has 2 identical alleles for a gene we call homozygous, and an organism that has 2 different alleles for a gene we call heterozygous. Mendel also came up with a hypothesis for how alleles interact. His idea was that if 2 alleles are different from one another, the allele that is seen is the dominant allele, and the one that is not seen is the recessive allele. To put this into context, if you are homozygous for the blue eye allele, your eyes would be blue. If you are homozygous for the brown allele, your eyes would be brown. When you're heterozygous, which means you have 1 brown allele and 1 blue allele, your eyes are brown. We would then say that brown is dominant to blue. Blue is recessive. Sometimes students will conflate the idea of something being common with it being dominant. Whether or not something is common has nothing to do with its relationship to other alleles. For example, let's look at these traits. On the top we see freckles versus no freckles. The dominant trait is freckles. If you're heterozygous, you will have freckles. You have to be homozygous recessive to not have them. But it's very common to not have freckles. Freckles are actually pretty rare. On the flip side, having normal pigmentation versus being an albino has a different relationship. If you're a heterozygous, you will be normally pigmented. If you're homozygous recessive, you could be an albino. Being an albino is extremely rare, so you can see that the thing that is common, no freckles and normal pigmentation, has very little to do with whether or not the trait is dominant or recessive. The last hypothesis was that sperm or eggs carry only one allele for each inherited characteristic, because allele pairs separate or segregate from each other during the production of gametes. This statement is called the law of segregation, and it's related to this idea of independent assortment. So if we were to put this on one slide and make it much shorter, we would say this. Mendel's hypotheses were that alternate versions of genes are called alleles, and they exist. Diploid organisms have two alleles for each trait, one from each parent. If they're the same, they're homozygous. If they're different, they're heterozygous. Alleles that can hide another allele are dominant, and alleles that can be hidden are recessive. And sperm and eggs are haploid, so they only carry one allele for each gene. Now let's apply what we've talked about to the experiment I introduced before. In this experiment, we started with true breeding parents, purple flowers and white flowers. We crossed them together to create an F1 generation where all of the flowers are purple, and at the end, when we cross F1s together, we get an F2 generation where 3/4 of the plants are purple and 1/4 of them are white. So why did this happen? Well, first, every diploid cell has to have pairs of homologous chromosomes. Remember that on those are what we call genes, and genes have a physical location on the chromosome. When we're talking about genes, we'll often assign them a symbol based on whether or not they are dominant or recessive. So for example, we like to use capital letters, like the big B here, to say that this particular allele is dominant to the recessive allele, which we assign a little b. This allows us to come up with what something's genotype is. So what is a genotype? A genotype is the combination of alleles that the organism has. So for example, if we were to look at the P gene here, we could say that there are two big P's. Therefore, the genotype of this organism at this gene is big P, big P. We can do the same thing for the other genes. That means that we have big P, little a, and big B, little b. We would also say that for P, this organism is homozygous dominant, because it has two copies of the dominant allele. It's homozygous recessive for the A gene, because it has two copies of the little a. And it is heterozygous, it has one dominant and one recessive for the B gene. Now we can apply this to our plants. If we cross purple and white together and we get purple, that tells us something about the relationship of these alleles. We talked about how dominant traits can cover up recessive traits, and recessive traits can be hidden. That means that purple is dominant to white. So we can define big P as the dominant allele purple, and we can define little p as the recessive allele white. So if I were to show you a random purple flower, what would you tell me about its genotype? Well, the phenotype of the plant is pretty clear. A phenotype is the appearance of a trait based on its genotype. The phenotype of this plant is purple, so we know that it has to have at least one big P. But big P, little p, the heterozygous state, and big P, the homozygous state, look the same, because big P is dominant to little p. So we don't actually know. We would have to write the genotype as big P dash. What about if I showed you this flower? This is a white flower. Do we know its genotype? Well, we know that the white flower is recessive to purple, and we assigned a little p to the flower. We also know that the only way to be white is to have two copies of the recessive allele, so we do know that this plant has to be little p, little p. Now, I told you in this experiment that we started with true breeding plants. So why do we know that these plants, this purple parent, has to be big P, big P? We know it has to be big P because it's true breeding. I told you, essentially. True breeding is going to mean homozygous. And we know that the little p plant has to be little p because that's the only way to be white. And I also told you it was true breeding. So now we can think about why are all of the plants purple in the F1? Well, they get one big P from their purple parent and one little p from their white parent. This makes them all heterozygous, and since purple is dominant to white, they're all purple. The F1 generation flowers can give one of the alleles that they have to each of their offspring. So they will give half of their offspring the big P and half of their offspring the little p. Normally when we think about this kind of thing, we can easily put it into what we call a Punnett square. Essentially what a Punnett square does is it takes each possible gametes and puts them on the x and y axis. One axis represents the possible sperm, and the other axis represents the possible eggs. We can then go through and add each of the letters to the box. So for example, all of the top ones will receive a big P from egg, and all the bottom ones will receive a little p from the egg. All the progeny in the first column will get a big P from sperm, and in the second column will get a little p from sperm. When we do this, we see that we get the same results that we saw before, 3 purple to 1 white. From a genotypic standpoint, we get the following ratio: 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. This is called a monohybrid cross, and this is the standard genotypic ratio. Our phenotypic ratio is going to be 3 to 1, 3 purples to 1 white. A cross between two individuals that are heterozygous for one characteristic is always called a monohybrid cross, and it's always going to have this same ratio: 3 to 1 for phenotype, and 1 to 2 to 1 for genotype. Crosses that involve heterozygotes for multiple traits have different names. The highest number that we'll deal with is going to be a dihybrid cross. In this case, the individuals that are mated in the F1 are going to be heterozygous for 2 things. While you can solve dihybrid crosses as a single event, you can also separate them into the 2 monohybrid crosses that they represent. This is a lot easier when you start doing practice problems, which are going to be very important for understanding this chapter. The reason you can get away with separating dihybrid crosses into the 2 monohybrid crosses they represent is because of the law of independent assortment, which states that alleles of a pair segregate independently from all other allele pairs during gamete formation. So I don't know about you, but I'm tired of using P's, so we're going to use cats. And we're going to use the following cats. We have a solid black cat on the left and a solid gray cat on the right. This difference, whether or not the cat is a dark color or a lighter color, is caused by a gene called dilute. The dominant version of dilute results in a full-colored black cat, but the homozygous recessive individual will be gray. Of course, there are some exceptions like this cutie, but we won't be thinking about them quite yet. So these are trait number 1. The second trait we're going to look at is whether or not the cat has stripes. This is controlled by the agouti gene, which determines the presence or absence of a coat pattern. What exactly those stripes look like is controlled by several other genes, but we won't be thinking about them quite yet. So that you can get an idea of what these traits look like together, the cat on the left would be a black cat that has a pattern, whereas the cat on the right would be a dilute cat that has a pattern. The reason the pattern shows up is because the agouti gene allows for color to be deposited into the hair in a stripe pattern. When the hairs lay down together, they create the illusion of stripes. As you might imagine, having stripes is dominant, and not having stripes is recessive. Now let's set up the experiment. We're going to start with a cat that is homozygous dominant for both traits. That means this cat would be black and have a pattern. We're going to cross that cat to a cat that is homozygous recessive for both traits. That means that this cat will be solid and gray. This is our parental generation. What is the F1 going to look like? Well, the cat on the left can only give dominant traits. and the cat on the right can only give recessive traits. This means that the cat that we get in the F1 is going to have one of each. It's going to be heterozygous for both traits, which means it's going to look like the parent on the left. It will have both a pattern and be black. Under normal circumstances, you should not do this, but we are going to be terrible home breeders. So we are going to interbreed our F1 generation. That means we're going to take two individuals who look the same and are heterozygous for both traits. So we want to ask a couple of questions. How many phenotypes should we get? What's possible? Well, our options are 4. We could get black cats, black tabby cats, which have stripes, dilute cats, or dilute tabby cats. There's a formula that we can use to determine the number of possible phenotypes, and that formula is 2 to the n, where n is the number of genes. We're dealing with 2 genes, so 2 squared is 4. The next thing we can think about is how many genotypes should we get? Each gene has 3 possibilities. For the dilute gene, we could be homozygous dominant, heterozygous, or homozygous recessive. That's what we would expect from a monohybrid cross. And we can also get the same thing for the other gene. We can use this formula 3 to the n because there are 3 possibilities for each hybrid cross, where n is the number of genes. This means that it's possible for us to get 3 squared, or 9 possible genotypes. Now let's go ahead and set up our Punnett square. To do this, we're going to need a lot more squares. So we're going to put the types of gametes that the first cat can make on the top line. Notice that each of these would be haploid. They have 1 copy of each possible gene. So this cat can give big D big A, big D little a, little d big A, and little d little a. Those are all the possible gametes this cat can make. The other cat is the same, so it's going to put its gametes over here, and we're going to populate that the same way. Big D big A, big D little a, little d big A, little d little a. Now we do the same thing that we did before, and we go ahead and populate each of the boxes with the different gametes from those cats. So for example, in this first box, this cat is going to get a big D and a big A from one parent and a big D and a big A from the other, making it homozygous dominant for everything. So when we fill everyone out, we get this. These would be all of the possible genotypes. Now to make it a little bit easier to see, I'm going to replace these genotypes with what the cats look like. Let's count up the total number of each type of cat. If we think about cats that look like the dominant parent, there are 9 of them. Here they are circled. If we think about cats that look like the black solid phenotype, there are 3 of those. There are also 3 cats that are dilute tabby, and there's 1 cat that's solid dilute. This gives us a 9 to 3 to 3 to 1 phenotypic ratio, and this is the standard ratio for a dihybrid cross. Now the genotypic ratio is a little bit different. So for example, we have 1 that's DDAA, all big, 2 that are 2 big Ds and heterozygous, and 1 that is 2 big Ds and homozygous recessive. This is the same 1 to 2 to 1 ratio as a modern hybrid cross, because basically that's what this is. Only one of the traits was heterozygous. Now, this is going to keep going. The genotypic ratio for a dihybrid cross is a mess. 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. I won't be asking you for this, okay? So you don't have to memorize it. But you are going to need to understand where these kinds of numbers come from.